The overall goal of this procedure is to detect detergent-sensitive interactions between membrane proteins or other proteins. This procedure requires one protein to be histidine tagged and overexpressed in the cells, while the other is a mic-tagged peptide.
This method can help answer questions in the field of biochemistry in protein-protein interaction studies. The main advantage of this technique is the ability to detect the interaction between membrane proteins in a detergent-sensitive way. The procedure is fast and easy.
Though this method can provide insight into membrane protein interactions, it can also be used to study weak protein interactions.
We first had the idea of this method when we wanted to check the interaction between the membrane proteins Sortilin and GLUT4, but attempts to co-immunoprecipitate them from the mammalian lysate remain unsuccessful.
To place an order for the peptide, select the target sequence of the peptide critical for interaction and add a mic epitope at the N or C terminus of the sequence. Once the peptide has arrived, prepare 100 micrograms per milliliter of working solution of the peptide in ultra-pure water. Seed 3T3L1 pre-adipocyte cells in 4 10-centimeter culture dishes and keep in the incubator. Next, transfect 2 culture dishes with the target protein tagged with 6X histidine and label as HisP, and the other 2 dishes with 2 control plasmids and label as WT. After transfection, leave the cells in the incubator for 48 hours to reach 80 to 90% confluency.